Hello everyone. So today let us try and understand what is extracellular matrix. Okay. So extracellular matrix is basically the connective tissue which surrounds the cell, okay, which is present in a tissue and an organ. Okay. For example, this is the cell and the tissue which is surrounding the cell or the connective tissue. This is the extracellular matrix. Okay. Connective tissue surrounding the cells. Now, what are the functions of extracellular matrix? Okay, first it forms the basement membrane of the cell, okay, on which the cell rests, helps in cell alignment, okay, it keeps the cells together, helps in the alignment of the cell, helps in cell proliferation, the various growth factors pass through it, okay, it helps in cellular proliferation, provides cushioning effect and protection to the cell, provides elasticity to cell in tissues like skin, blood vessels, ligaments, lungs. It also provides strength and shape to the tissues, and it helps in formation of thrombus in blood vessels. Okay, so these are the functions of extracellular matrix. Now, how are extracellular matrix classified? Okay, so they're classified based on the function. Okay, they can be structural proteins like collagen, elastin, fibrillin. So these are all structural extracellular matrix. Okay, and they provide structure to the cell. Okay, they help in protection, provide protection to the cells. They provide the basement membrane and also elasticity. Next, it can also be specialized proteins like laminin or fibronectin. Okay, so for example, you can see over here, this is the cell. The surrounding tissue is called the extracellular matrix, okay, or the connective tissue. So this is the collagen. Okay, this is the type 4 collagen on which the cell is resting. So it provides the basement membrane on which the cell is resting, okay, and helps in various cell alignment and protection and all those things. Now laminin, it helps to connect the type 4 collagen to the cell. Okay, so the cell is connected to this type 4 collagen, okay, with the help of laminin. It binds the cell and type 4 collagen together. Okay, so this is specialized type of extracellular matrix. Next we have is fibronectin. Okay, so fibronectin, basically it helps the cells to keep together or cell addition. It also helps in cell to cell communication and migration. Okay, and lastly we have is proteoglycans, which are nothing else but glycosaminoglycans with polypeptide backbone. Okay, so let us see what proteoglycans are. Okay, so you can see over here, see, proteoglycan is protein backbone plus glycosaminoglycan. Okay, so this is the structure of proteoglycan, like a bottle brush. Okay, so this is the core protein or the protein backbone, and to this you have is glycosaminoglycans which are attached. So these small green dots, these are the glycosaminoglycans. Okay, so if you see one section of this green section, you can see this is the glycosaminoglycan. Okay, so proteoglycans are protein backbone with glycosaminoglycans. Okay, now this glycosaminoglycans is made up of repeating disaccharide unit. Okay, so repeating 2 units, so which is uronic acid and amino sugar. Okay, so this is the uronic acid. Okay, so this is the uronic acid and this is the amino sugar. Okay, so amino group is over here, so this is the amino sugar. So we have a repeating unit of uronic acid, amino sugar, uronic acid, amino sugar, and so on and so forth. So this is known as glycosaminoglycans, and these glycosaminoglycans are attached to this protein backbone, and thus it becomes a proteoglycan. Okay, now what are the functions of proteoglycan? So they are found in literally every tissue. They form the major component of extracellular matrix. They interact with structural extracellular matrix like collagen and elastin. They bind with growth factors and modulate cell growth. They interact with adhesive proteins like fibronectin and laminin. They are negatively charged, hence attract cations like sodium and also water molecules, which leads to their viscosity. Okay, so that is why proteoglycans are viscous in nature. Depending upon the glycosaminoglycan composition, they serve important functions in bone, cartilage, synovial fluid, vitreous humor of the eye, blood vessels, cornea, skin, etc. Okay, so this proteoglycan, what is proteoglycan? These individual glycosaminoglycans, okay, which glycosaminoglycans are present in skin like dermatan sulfate, in synovial fluid like hyaluronic acid. This we have already discussed in the chemistry of carbohydrate lecture. Okay, moving forward, so let's talk about collagen. Okay, so collagen is an important structural extracellular matrix. So let us see how collagen is synthesized in our body briefly. Okay, so collagen is a structural extracellular matrix. It can be in the form of a rope or a mesh. Okay, so rope is more like a fiber and mesh is more flattened out. Okay, so it has a triple helical structure. Now based on its type, if it's like a rope or a fibril forming, it is seen in skin, bones, tendons, blood vessels, cornea. So here type 1 collagen is seen. Okay, so type 2 collagen also is a fibril type or a rope type and seen in cartilage, intervertebral disc, vitreous humor. Okay. Similarly, the type 4 collagen is more of a mesh forming or a network forming. So this is seen in basement membrane. So this is where the cells they rest on this type 4 collagen, okay, which is type of a structural extracellular matrix. So the basement is formed by type 4 collagen, which is a mesh forming type of collagen. Okay, so and we also have this fibril associated type of collagen, collagen 9 and 12. Okay. So if you look at the structure of collagen, it is a triple helical structure. It has repeating sequence of glycine XY, again glycine XY, glycine XY. Okay, so glycine is the only amino acid that is small enough to accommodate in a triple helical structure. Okay, so the glycine is the smallest amino acid that can be easily accommodated in the triple helical structure. The X is usually the proline and Y is hydroxyproline, or it can be Y also can be hydroxylysine. Okay. Now let us look at the structure of collagen. Okay, so this is the triple helical structure. Okay, you can see 3 fibers which are intertwined together. Okay, blue one, green one, and red one. Okay, they form a triple helical structure. So how this triple helical structure is formed? Okay, so this triple helical structure, it has a staggered appearance. Okay, so this staggered appearance is seen as light region and dark region in the microscope. Okay, so we can see that the arrangement is, we have a dark stained region as well as a light stained region because of this staggered appearance of collagen fibers. Okay, now let us see how collagen is synthesized. Okay, so the collagen is synthesized. The gene for synthesis of collagen is either the fibroblast or the osteoblast. Okay, so osteoblast or fibroblast gene, they undergo transcription and form an mRNA of collagen. Now this mRNA, it goes to the ribosome. So in the ribosome, we have formation of pre-pro-alpha protein. Okay. Now this pre-pro-alpha protein which is formed in the ribosome, it has a special sequence at N terminal. Okay, or N signal is there. Now this special sequence directs this pre-pro-alpha protein to endoplasmic reticulum. Okay, so when it goes into the endoplasmic reticulum, this pre-pro-alpha protein is broken down into pro-alpha protein and the N signal. Okay, so the purpose of this N signal was to direct it to endoplasmic reticulum. So when it comes into the endoplasmic reticulum, then this signal is lost. Okay. Now what we have is pro-alpha protein. Now this pro-alpha protein undergoes hydroxylation of proline and lysine amino acids. Okay, so the sequence of amino acid is very simple. Is the simplest of sequence is present in collagen. Glycine XY, glycine, proline, hydroxyproline, or hydroxylysine is there. Okay. Now this proline or lysine undergoes hydroxylation Okay, they undergo hydroxylation because so that bond formation is easy. Hydrogen bond formation is easy, and hydrogen bond is necessary for cross-linking. Okay, so proline and lysine amino acids, they undergo hydroxylation. Why? Because they want to form hydrogen bonding. Why? Because hydrogen bonding leads to cross-linking of the fibers, which leads to structural integrity. Okay, now collagen is a structural type of extracellular matrix, so it needs to be very strong. Okay, so to maintain its structural integrity, you need to have cross-linking of fibers. This cross-linking is brought about with the help of hydroxylation of the residues like proline and lysine. Okay, so now this hydroxylation occurs with the help of vitamin C, molecular oxygen, and iron. Okay, all these three are essential for hydroxylation of proline and lysine. So in other words, if there is deficiency of vitamin C, the structural integrity of collagen will be compromised. Why? Because hydroxylation of proline and lysine will not be good. Okay, hydroxylation will be poor, hence the structural integrity of collagen will be also poor. And if the structural integrity of collagen, which is the basement membrane, is poor, so it can lead to breakage of the collagen. And in the blood vessels, which has the basement membrane type of collagen, it can lead to bleeding, which is commonly seen in vitamin C deficiency or scurvy. Okay. Similarly, we also have is glycosylation of hydroxylysine. Okay, carbohydrate also is added, which is glycosylation of hydroxylysine. Now, after the hydroxylation has occurred, we have three pro-alpha chains which come together and they form a triple helix. Okay, so three pro-alpha chain, they come together, they form a triple helix like this, and it is known as pro-collagen. Okay, so this pro-collagen has a central triple helical structure with a C terminal and an N terminal. Okay, so this is known as pro-collagen. So in the endoplasmic reticulum. So what is happening in the endoplasmic reticulum? First, the pre-pro-alpha is coming. Okay, then the N signal is lost, so it forms this pro-alpha protein. Now this pro-alpha protein, they undergo hydroxylation of proline and lysine. Okay, and three pro-alpha protein come together and form a triple helical structure or a pro-collagen. Okay, now this pro-collagen goes to the Golgi apparatus. The Golgi apparatus, when it goes to the Golgi apparatus destines it or makes it extracellular, okay, or tags it for delivery to outside the cell. Okay, once the pro-collagen goes to the Golgi complex for expulsion into the extracellular medium, when it comes out into the extracellular space, then C and N terminal of pro-collagen is lost or is cleaved, and what you get is tropocollagen. Okay, so this pro-collagen, this C terminal and N terminal is cleaved, it is broken down, and what you get is only tropocollagen. So this occurs outside the cell, extracellular. Now once you get this tropocollagen is arranged in this staggered fashion, okay, and forms as collagen fibrils. Okay, so these collagen fibrils, again, they form cross-link with lysine residues with the help of lysyl oxidase, and this also gives a very strong structural integrity to collagen. So this is how the collagen is synthesized, right from the gene, which is osteoblast or fibroblast, to formation of a staggered appearance extracellularly. Okay. Now let us look at some of the defects briefly, try to understand what the defects of collagen are, or collagenopathies. Okay, so collagenopathy is basically a defect in the synthesis of collagen due to various mutations. Okay, so first is Ehlers-Danlos syndrome. So Ehlers-Danlos syndrome is a type of collagenopathy in which there is deficiency of lysyl hydroxylase. Okay, so if lysine or lysyl hydroxylase, that means lysine is not getting hydroxylated, or hydroxylation if is poor. So we have seen that if hydroxylation is poor, cross-linking of fibers will be poor, and the structural integrity of collagen will be poor. Similarly, N-procollagen peptidase. So the N terminal is lost. If peptidase N procollagen peptidase is absent, then again the N terminal will not be broken down. Then there is a change in the sequence of amino acid in fibril forming collagen, okay, or type 1, type 4, type 5. So if the sequence of amino acid is changed, then also the collagen will become weak. Okay, so the weak and collagen becomes easily degradable. So all because of these problems, these mutations, okay, what happens is skin is extensibility is there, highly extensibility skin is there. Okay, you can pull the skin. Skin is very fragile. Okay, it bleeds easily or it's broken easily. Joint hypermobility is there. Okay, because the collagen is weak, so joint hypermobility is there, and the collagen lining the blood vessels also is weak, so it can lead to rupture of the blood vessels and bleeding. Okay, so this is what happens in Ehlers-Danlos syndrome. Next we have is osteogenesis imperfecta. Okay, this again is type of genetic mutation in which the glycine is replaced by a bulky amino acid. Okay, so we have seen that in the triple helical structure, glycine is the smallest amino acid which easily fits in the triple helical structure. Now if glycine is replaced by a bulky amino acid, then the triple helical structure will be little abnormal. Okay, it will be weak, it will not be of the same tensile strength, and this is known as osteogenesis imperfecta. Okay, so here again we have is the bones become fragile, hearing loss is seen, also the sclera becomes blue. Okay, as you can appreciate over here, a blue sclera, and the patient also has various lung complications. Next we have is scurvy, that we all know. It's because of vitamin C deficiency. In case of vitamin C deficiency, hydroxylation is affected. There is no hydroxylation. Cross-linking of collagen fibers is also defective. As a result, the collagen which is formed is very weak and easily broken. So this can lead to bleeding gums, it can lead to bruises, it can lead to weak bones and anemia. Okay, so we will discuss these again when we discuss individual disorders. So lastly we have is alpha-1 antitrypsin deficiency. In case of alpha-1 antitrypsin deficiency, the inhibition of elastase is lost in the lung. Okay, so in case of normally alpha-1 antitrypsin, what it does is it inhibits the enzyme elastase. Okay, so elastase is a proteolytic enzyme which breaks down the tissue. Okay, so we do not want an overactivity of elastase. If the elastase is overactive, it will lead to breakdown of the lung parenchyma, which leads to ultimately emphysema. Okay, so alpha-1 antitrypsin, it breaks down or it inhibits this elastase enzyme. So as a result, it protects the emphysema. Okay. Now what happens is if there is decreased alpha-1 antitrypsin or deficiency of alpha-1 antitrypsin, then the elastase enzyme becomes unchecked and leads to lung destruction. And smoking leads to a defect in alpha-1 antitrypsin. As a result, inhibition of elastase is also decreased. There occurs overactivity of elastase, which leads to lung parenchyma destruction, and we have is emphysema. Okay, so we will again look at these disorders in more detail when we talk about individual disorders. Okay, so I hope you have understood what is extracellular matrix, the types of extracellular matrix, the classification, and what is collagen and synthesis of collagen, and types of collagenopathies. Okay, so thank you very much. Thank you.